Okay, our next speaker will be Véronique Hamel, and her talk is titled Genetic and Environmental Backgrounds Constrain the Course of Evolutionary Rescue by Compensatory Mutations. I think I need a cup of coffee, sorry.
So, hi everyone. Today I will talk to you about how genetic background and environment can constrain the course of evolution. This project has been done when I was undergrad student, and I am now a master student in Landry's lab at Laval University in Quebec City, Canada. So I really want to thank GSA to give me the opportunity to give this talk, and also for the travel award. First of all, as we all know, today with the new technology, as new generation sequencing, we are able to know exactly which mutation in which gene is causative for disease, for a genetic disease. This knowledge give us the opportunity to be able to look for new treatments that will target specifically those genes. But even if we have this knowledge, there is a big challenge that remains. For example, for disease where they have loss of function for one gene, because for this, the cells have, the treatment have to pass away the fact that there is a loss of function for one protein in the cells that affect the growth of the cells. Even more, there's a more highly level of challenge. So for the fact that the disease and the treatment can be highly dependent on the genetic background and the environment. In this study, they have a case where they found in different people in the world mutations that are causative for a disease, but the patient didn't present the different symptoms. And these cases are really important because they probably have other mutations that give them the possibility to be insensitive to the disease mutations. And with that, we will be probably able to develop new treatments. These studies give us to ask ourselves different questions, such as can secondary mutation rescue cellular function, and can it restore the phenotype, and is it dependent on the genetic background and the environment? To answer those questions, we have decided to use a yeast model for a human disease, the Wiskott-Aldrich syndrome. This disease is a rare primary X-linked immunodeficiency and a blood platelet disorder. The patient will present diverse symptoms such as skin eczema. And for now, there is only one treatment available, so the bone marrow transplant. This disease is caused by deleterious mutations in the gene, the protein coding gene WASP. By using this yeast model, that carried a deleterious mutations, we have let emerge some compensatory mutations. So by compensatory mutations, I mean secondary mutations that are able to rescue the phenotype of the cells for the deleterious mutations. And the compensatory mutants that I will do is the cells that carry those compensatory mutations. In our work, we had identified the different compensatory mutations by sequencing. In yeast, the functional homolog of WASP is LAS17, and it is an essential gene that is involved in actin assembly. The allele of LAS17 that carried the same mutation as the human gene that provoke Wiskott-Aldrich syndrome is LAS1741, and it is a thermosensitive allele. So in yeast, the cells are able to grow at 22 degrees, but not at 37 degrees. Also, we know now by using different methods such as Western blot and protein complementation assay that LAS1741 protein is absent at 37 degrees. So that means that the cell have to pass throughout this to be able to compensate the complete loss of function of these genes. To be able to answer our question, is it dependent on the genetic background and the environment, we have decided to use two different strains, so BY and RM, that both carry the thermosensitive allele, and two different carbon sources that glucose and galactose that are metabolized differently by the cell. So for our experimental evolution, we have let the cell grow at 22 degrees to accumulate random mutations, and after we had select the compensatory mutants at 37 degrees that are represented here by the white dots. Typically, those cells carry just one compensatory mutation. With this experiment, we were able to accumulate different information, such as compensatory mutation rate, and we had also select 100 mutants to sequence them. If we check the compensatory mutation rate, what we are able to see and compare them between the different combinations of conditions for the combination of genetic background and environment, we are able to see that they are really different between the different conditions, and there is no tendency for one genetic background or another, or for one carbon source or another. So we are able to see that compensatory mutation rate is dependent on the genetic background and the environment. By sequencing, we were able to identify which compensatory mutation type we had. So we have found 4 different types. We have found deletion, SNVs, so point mutations, that represent most of the compensatory mutation we had found, and we can see that there are a lot of variations. And we also found aneuploidy and insertion. Those two cases are specific to one genetic background or another. So aneuploidy, we only found it in RM, and insertion only in BY. So those information give us the ability to say that compensatory mutation type is dependent on the genetic background and the environment. By sequencing, we were also able to identify in which genes we found compensatory mutations, and they are represented on this figure. So here, to explain you a little bit, the big circle represents the different combination of conditions. So for example, BY in glucose. The color of the nodes represent the function of the genes. The link between the different genes represent the protein-protein interactions. So what is interesting on this figure is that most of the genes that carried a compensatory mutations are interactants of LAS17, so they are implied in actin assembly or disassembly. But there are 3 of them here in orange that are not interactants of LAS17, but they are interesting too because they are involved in the same signaling pathway, so the calcium calmodulin pathway. What is interesting too is that we can see that some compensatory mutation solutions are more specific to one background, to one conditions. For example, RM in galactose with ARB3. And for CAP2, for example, it is a generic solution that is found in every different combination of conditions. Because we want to validate our candidates, we have done different experiments. The one that I will present you is the one that we think about loss of function of the genes. So for validate that, we have done deletion of the entire genes, and I present you here 3 examples that were validated. First of all, if I explain you a little bit the figure, here we found the control. So the wild type strain in both different genetic background can grow at 37 degrees, but not the one that carried the thermosensitive allele. But for those ones, when we delete, for example, BSP1 or trienphylline 1, we were able to see that RM is able to compensate or rescue its phenotype at 37 degrees on both carbon sources and for the two cases. And we see also when we delete CNB1 for BY, the same thing. So there is compensation for it at 37 degrees on glucose. So those cases are important because by deletion, we can also target them by different drugs. And that's what we have done for CNB1 because we know our drug, cyclosporine A, that is able to target calcineurin that contains as a subunit CNB1. And we have tried it. So here again, when we don't put the drug in the media, the control that carried the thermosensitive allele are not able to grow. But when we put the cyclosporine A in the media, BY in glucose is able to rescue its phenotype. So I think it's a really cool result. To conclude, how those results together, we can say that compensatory mutation rates are enriched in last 17 protein-protein interaction network. And for a more general conclusion, that probably means that protein interaction partner may be prime therapeutic targets. And we can also conclude that the paths that evolution can take to compensate for deleterious mutations are influenced by the genetic background, the environment, and the interaction of both. For the medical field, that means that therapeutic targets could differ from one patient to another, and that's really important for precision medicine. For future research, I am really interested to understand how the divergence between BY and RM for their cellular network diverge for giving us different compensatory paths. So I want to thank all the members of my lab, more particularly Marie, Isabelle, Alexandre, and Christian, all the funding grants, and thank you for your attention. I am ready to answer your questions.